Welcome all, and thank you for joining today's session. My name is Michael Downey, and it's great to be on board for another AWRI webinar. This is the penultimate session for the year, and as with previous years, the final session scheduled for next week will be a Bureau of Meteorology Climate Outlook for Vintage 2020. This week, however, takes a look at soil health, and more specifically, how to monitor the health of your soil. But before we get to today's topic, some quick reminders for anyone new to AWRI webinars. To provide a comment or to ask a question, please open the Q&A section of the webinar, type in your question and click to send it through. A reminder, this webinar is being recorded and it will be available to view via the AWRI's YouTube channel. For those of you that have just joined, welcome. Today's AWRI webinar takes a look at soil health, and it's a great pleasure to welcome today's speakers. Dr. Mark Krstic is the General Manager, Business Development at the AWRI and has extensive experience in wine industry RD&E, having worked at Wine Australia, Victorian DPI and CSIRO before landing here at the AWRI. Mark has also recently co-authored a fantastic new book titled Healthy Soils for Healthy Vines. His co-author, Emeritus Professor Robert Wright from the University of Melbourne, is also joining us today and has generously agreed to take part in today's webinar. So we'll hear a little bit more from Robert later in the session, but it's fantastic to have both Mark and Robert's expertise on hand. And with that, let's jump in and make a start. So Mark, over to you.
Thank you, Michael. And yeah, it's an honour to be doing this today with Robert. And I'll bring Robert in. Robert's got some time constraints today, but I'll bring him in at certain times during this particular webinar. So my topic today, soil health checklist, what to measure to assess soil health. So let's get going. So the talk outline today. So I just want to start off with a little bit of background, talk a little bit about soil health and what do we mean, and then look at the approaches to assessing soil health, both indirect and direct assessments. Look at how we then go about interpreting soil health analytical data, introduce to you the concept of a minimum soil health data set for vineyards, and look at the choices we have when establishing either new, established, or actually reworking vineyards, and then sort of summarise some of that sort of stuff there. So look, Bob's only available for the next 30 minutes, so I'm going to bring him in a little bit earlier, about halfway through the presentation at some point if you're wondering where Bob comes in. Okay, the background in terms of today, and I guess why I'm here today to talk to you, is that Robert and I have recently co-authored a book which was released in September this year, as Michael said, called Healthy Soils for Healthy Vines. And that book, is available now through CSIRO Publishing. And really what I'm focusing in on today in this particular webinar is really a focus on chapter 4, where we talk about assessing soil health in that particular book. But there's much more information in the book around soils, but also around assessing soil health. So when we talk about soil health, what do we mean? So the commonly used definition that we have at the moment is, in terms of soil health, is it really relates to the capacity of a soil to function within ecosystem boundaries to sustain biological productivity, maintain environmental quality, and promote plant and animal health. What does it mean, in drilling down a little bit further? It's really a relative judgment made according to our expectations with respect to fitness for use. Dynamic can be influenced and changed compared to soil quality, which is inherent. So it's really important to understand both those inherent and dynamic properties of soil. And understand that soil is obviously part of the farm infrastructure, representing the natural capital that we see in a vineyard. Soil health, it's really important for vineyard attributes around water retention, soil structure and resistance to compaction and erosion, nutrient cycling and plant nutrient uptake, organic matter management and biological diversity. When we really break down soil health, we're really talking and really getting into understanding, we're talking about 3 main elements. And those are talking about the soil physical properties, the soil chemical properties, and those biological properties. And, really dissecting and trying to understand, what do we measure? What are the attributes we measure around those that give us some indicators around what we refer to as soil health? When we come to assessing soil health, there have really been two main approaches. The first is a more indirect method, and it incorporates the practices of sustainable wine growing. And that refers to really practices that are both economically viable, socially responsible, and environmentally sound. The other one, which we're probably more familiar with, is the direct method that focuses on key indicator properties of the soil or the vine, and where values are then compared against benchmark values or optimal ranges, and the trends are monitored over time. And again, this infers that there's an understanding of both the inherent and dynamic soil properties. So firstly, let's look at the indirect methods. So many countries around the world now in the wine sectors have sustainable wine growing programs. So we know that there's the Sustainable Wine Growing New Zealand program. There's certainly ones in California, South Africa, and also here in Australia, we have the Sustainable Wine Growing Australia program, which is now the single national sustainability program that we have here and that we're really pushing out. And it's really only commenced from July 1 this year onwards. Within their annual workbook, there's a land and soils chapter, and there's a range of different parts that I guess vignerons are asked to sort of rank themselves in terms of where they sit, from soil management plans right through to the fertilizer and soil additive storage and handling. I've highlighted two in red there just to sort of give you a little bit more detail on those. So talking about soil nutrient analysis and organic matter. So lots of words here, but I just really want to say that, under the Sustainable Wine Growing Program here around soil nutrient analysis, it really is it's a self-assessment system here to say, well, where do I sit? Am I a zero, where there's been never any soil analysis performed or any soil analysis records for the particular property, right up to, that they've done a soil management plan and regular soil analysis, and they've trained their staff on a regular basis at level 4. So people are able to self-assess and see where they sit on that spectrum of conducting soil nutrient analysis. If I move now to just look at the organic matter self-assessment area, again, you can see that 0 relates to the status of soil organic matter in the vineyard is unknown, and up to 4 where, they understand that they're measuring soil organic matter and really working on, limiting cultivation in certain areas and really working on, I guess, what they can do in terms of compost and mulch addition to the vineyard to really improve it. So again, it's a self-assessment tool, a little bit easier. It doesn't involve any necessarily, I guess, doing any measurement per se, but just recording on where they sit on those particular scales. If I switch now to looking at approaches for assessing soil health and looking at the direct methods, generally there are 3 different scenarios that may confront a producer when considering options to assess soil health. You might be in a situation where you've got a brand new site, it's a greenfield site, never had grapevines on it, and so there's a different sort of frame of reference that you're looking at in terms of thinking about your assessment of soil health in that particular situation. You might be in an established vineyard where you've got grapevines and trellis in place, and again, it's a different sort of set of indicators you might be thinking of here. Or you may have an existing vineyard where you're looking to actually redevelop that vineyard, maybe pull it out completely and rework and replant with a new variety, put a new trellis in, whatever, but there's different considerations that I'll touch on later on in the presentation about those. I guess the key thing is each option requires a different approach and consideration of which soil health indicators might be selected for assessment in those particular scenarios. In relation to, when we're looking at assessing soil in a vineyard and the soil health properties, really we can't take all the soil from that vineyard and submit it to the laboratory and come out with a result for what it is. We're taking representative samples across that vineyard that then reflect what we're actually seeing in the vineyard. So it's important to understand those sampling issues. Now, I guess traditionally, in many other parts of Australia, there's been an approach around using a 75 by 75 metre grid pattern across a new property and taking either pits or cores from that and actually having those submitted for analysis. I think, with the advent of precision viticulture tools and especially ground penetrating radar approaches, the ability to map that soil and that variability across that soil and be a little bit more strategic about how we place those soil we sample that soil across that vineyard is really important. And I think that's a real advance in the technology. You know, it's been around now for 10 or 15 years, of course, but we need to, you know, really embrace that, and in terms of how we select our sites for soil analysis. I think prior knowledge of that spatial variation does allow for effective location of sampling via either soil pits or other soil collection methods, you know, using either a mechanical auger or a corer to take soil samples across that pit. I guess I just wanted to pause here for just a second, really, and just say, look, you know, I know a lot of people do soil pits, and I certainly strongly encourage it. And there's a lot of things that can be done. I mean, we tend to race down and look at, you know, taking samples and sending them off for analysis and getting the numbers back and then trying to understand what they mean. But I think it's really important just to pause here that, you know, if you've got an established vineyard and you're doing soil pits, really, these are a great opportunity to see how the root systems in the soil are distributed, and understanding where there may be some points or problems around soil compaction. It also, you know, there's a lot of existing literature that is out there also within these existing wine growing regions on the general soil types you're likely to see across the region, and some of those physical and chemical properties even. So, you know, understanding, you know, where your soil fits within the region from those existing soil maps, and understanding, you know, some of those issues there. So I just wanted to really reiterate that while we're going to be talking a lot about measuring these attributes from this point onwards, that a lot can be gleaned from, you know, just having soil pits in the vineyard, being able to really make some of those assessments. And, you know, if you've got the skills to also, you know, do some of those basic things around soil texture analysis. Okay, so, but moving on to the more, I guess, analytical approach to measuring some of these different physical, chemical and biological attributes, you know, the choice of those depends on, you know, ensuring that they're sensitive to changes in management and or environmental conditions, that they're easy to measure and interpret, that the measurement technology is robust and repeatable, and I'll come to this after we talk about the actual attributes as well, that the attributes are reversible so that both improvement and degradation across time can be monitored. And it really is important to understand the pros and cons of each measurement. So this is where we're really going to get into a little bit of detail in relation to the different properties under each of those categories of physical, chemical and biological attributes here now. So I'm going to walk through, and what I'm going to do here is pause after the end of each of the broad categories of physical, chemical and biological, and then we can engage and talk. You know, if there's any technical questions, I can make sure Bob's talents are utilised in this particular part of the presentation. So bear with me as we go through this. So I've got a table here, and there's a number of slides that cover off on the physical attributes here. So first of all, I want to talk about, you know, soil texture. It's a really important for the initial characterisation of the soil. It's a crude indicator of water available capacity, which I'll touch on later as well. In terms of the cons, it's not suitable to monitor over time because it's unlikely to change. So, you know, that's the first one. Aggregate stability, it's a good indicator of soil structure and should reflect management changes, such as minimising mid-row cultivation and using cover crops, and it can change quickly. In terms of cons, there's none except for some of the measurement limitations, and some of those require obviously using wet sieving and specialised equipment. The third soil physical property is soil strength. So indicates the soil's tendency to compact when wet and resist penetration by roots when dry. So I guess the con here is in terms of the soil spatial variability can be a problem. And often, you know, these are really measured using a penetrometer, and it's important to make sure that the soil moisture content is at the similar level when you're taking those particular measurements as well. The fourth soil physical property that I want to talk about is bulk density. It's inversely related to a soil's porosity and directly related to soil strength given at a given moisture content. So that's, you know, the pro. The con, it's feasible for only really the surface soil, unless you've got a soil pit and you can actually remove an undisturbed soil sample from. Looking at porosity and airfield porosity indicates the soil's capacity to hold water and gases. Airfield porosity indicates the soil's level of aeration. Some of the negatives are the same limitations as for bulk density, but the airfill porosity is difficult to measure. Surface crusting indicates the ease of water infiltration, and it's related to water stability of surface aggregates. Issue there though, I guess the negative is primarily a problem when soil is dry and may not be obvious when wet. And so the quantitative measurements with a penetrometer can give you an idea there, or it's really around visual observation when dry as well. The last couple of soil physical attributes, infiltration, obviously important for understanding soil structure and the hydraulic conductivity. But at the same time, the rate depends on soil moisture concept, and there's considerable variability across an individual site. Last one here, available water capacity. It's the volume of water per unit soil depth and is notionally available between field capacity and wilting point. And so I certainly use this a lot when estimating plant available water or readily available water based on texture results. So can be useful as well. So what I might do there, because I've gone through probably about 8 different attributes, I'd like to pause if I could and just bring in Bob, just to sort of mention, Bob, are there any other elements you really wanted to discuss around those soil physical attributes, and give people an opportunity to ask questions at this particular point as well?
Thanks, Mark. Just one point with regard to available water capacity, which is different from the plant available water because, as you mentioned, available water capacity is volume per unit depth of soil. But of course, if you have variations in soil depth and rooting depth of the vines in particular, then you multiply the AWC, the available water capacity, by the depth. And this gives you the plant available water. So even for soils that have the same available water capacity, if you've got very different rooting depths, you can get very different plant available water. That's just a small point to make.
No worries. Thanks, Bob. Are there any questions from attendees here on the webinar about any of those things that we've talked about thus far? And I'll pause here for a little bit.
Yeah, no questions as yet, Mark. Just a reminder to the audience, if you do want to ask a question, just open the Q&A button on the webinar toolbar, type them in and send them through. But at this stage, we do not have any questions, Mark.
I might push on in the interest of time then. So let's move on to soil chemical attributes, and probably no surprise here for many of these. So I'll start off with the first one, obviously soil pH and the pH buffering capacity. So really important for understanding the availability of nutrients in the soil, and I've included that graph on the right-hand side of that slide there to really understand more about that. And I think I won't really go into too much detail because I think that's quite self-explanatory. Other ones around soil organic matter. Of course, soil organic matter is really important for supporting the microbial growth, and it's certainly important in nutrient recycling and soil structure. No real negatives around that one, except for qualifications about measurement. Most meaningful for soils really in that top 0 to 15 centimetre layer, and it changes slowly. Total soil nitrogen. Most nitrogen in soils is in those organic forms, and the C/N ratio indicates the likelihood of nitrogen mineralisation as well. Some of the negatives is not directly related to mineral N, but can be a surrogate for potential mineralisable N, and does change slowly. Continuing on, looking at available phosphorus. Obviously, some of the pros are it's an indicator of plant available phosphorus and can quickly change and more evident than total soil P. The negatives, though, are that the values very depend on the method of measurement. So it's really important to, if you are looking at soil phosphorus, to make sure that you're comparing numbers from the same method and the units are the same, and I'll touch on that a bit later on. Cation exchange capacity affects, cation retention and structure formation, and can change with soil organic matter. No no real negatives here, but just remember that the cation exchange value depends on the pH at which the measurement's taken in that particular situation too. On from that, exchangeable cations, really the sum of the calcium, magnesium, potassium, and sodium, and measures the store of available basic cations. And understanding the percentages of those cations from those different elements is really important in understanding some of the soil stability issues as well. Electrical conductivity, a surrogate for total soluble salts, and it certainly can change rapidly depending on irrigation, water, rainfall, and leaching, and really important in understanding, I guess, root zone salinity for us, and pretty easily measured using a handheld EC meter, 1 to 5 sort of water suspension, or it can be measured in the saturated paste as well. And understanding those exchangeable sodium percentage is really important too, again, just in knowing whether our soils might be exhibiting some acidicity problems. And it's derived from exchangeable cation measurements, and that's a good indicator, like I said, of soil acidicity. So I might pause there again and bring in Bob, just to say, Bob, are there any technical elements that I might have left out there that you wanted to raise around soil chemical attributes?
No, I think you covered it very well, Mark. The only thing I would emphasise that in a lot of Australian vineyard soils, particularly in southeast Australia, the exchangeable sodium percentage is very important in terms of determining whether you're likely to have a stable structure or not. And coupled with that exchangeable sodium percentage is the ratio of calcium to magnesium. Preferably that ratio should be greater than 2, and if it's less than 1, that's calcium to magnesium, then it exacerbates the problem of structural instability.
Thank you, Bob. Any questions at that point there?
Yeah, so we do have a couple of questions that have come through. First one here is a question about how often soil samples should be taken to check for changes over time.
Did you get that, Bob?
Yes, yes. Well, that depends on whether you're measuring something which is changing fairly rapidly or not. But things related to the cations, you probably don't need to monitor that more than every 3 years or something like that. But if you're looking at some of the biological properties, then you might need to sample a bit more frequently. And with respect to the physical properties, then to the extent that the stability of structure is dependent on the exchangeable sodium percentage and so on, well then that would determine how often you might want to look at the soil physical condition, which might be every 3 years.
Thanks for your comments there, Bob, and your question, Jackson. Did you want to make a comment as well, Mark? No, we've got another question around permeability and the falling head test method. Did you have any comments around that, Bob?
Well, that's something which is applied to measuring how fast water moves out of a hole which you dig in the ground, usually with a coring device, and you can study the subsoil permeability in that way. It's quite a valuable technique. But in many cases, you can get an idea of soil permeability from examination of the profile pits in the first place. This is what I like to do, because when you have a profile pit, you can look at the faces of the pit, which should be relatively undisturbed, and you can get an idea of whether you've got a compacted subsoil or not, and whether that might present problems from the point of view of permeability. The other thing is that you can tell a heck of a lot from changes in colour in the soil profile from a profile pit. And when you see the phenomenon of mottling, which is orange-red mottles within a pale soil matrix, then you know that you've got at least temporary waterlogging at times of the year, and this indicates poor drainage. So there are ways of assessing soil permeability other than having to use a falling head technique, for example.
Okay, great. Thanks for your insights there, Bob. No more questions at this stage, Mark, so I think we can continue on.
No worries. So let's push on. So the last category in terms of you're looking at soil quality attributes is really those soil biological ones. And look, I guess I wanted to just introduce this and say, look, while we know certainly a lot more about the soil physical and chemical attributes, there's been 150, 170 years of collective research in those particular areas. The soil biological area is one that's sort of rapidly we're learning a lot more rapidly, and there's been a lot more effort over the last perhaps 30 years in this area relative to the other two. So just to I guess give that a bit of context. But in terms of soil biological tests, they fall broadly into two categories. One around measurement of the numbers, activity and diversity of soil organisms and their related biochemical processes. So these tests are primarily focused on assessing how well the soil functions as a natural ecosystem and supports healthy vine growth. An implicit assumption though in this is that healthy biological activity also makes the grapevine more resistant to pests and diseases. The second one, which we'll touch on a little bit later, but it's not really probably that important for this particular webinar, is complementary to that first category, the measurements of pests and diseases that can adversely affect vine growth and may cause death. And these are things where we're looking at things like phylloxera and nematodes that might attack the roots and cause ill health to the vine. I guess, a lot more interest in, understanding that soil food web. So understanding things from, the first trophic level right through that soil food web, understanding the microbiological attributes, the nematodes, arthropods, earthworms, and those sorts of things within the soil is really important to understanding or getting an indicator to how well the biology of the soil is functioning and the health, and therefore the overall health of that soil. I think it's also important to note that obviously getting the right physical and chemical attributes is also important for making sure that we've got good biology in our soils as well. So let's go through some of these. And in terms of some of the soil biological attributes, obviously soil organic carbon and labile carbon is really important. It's important, labile carbon is an easily decomposed component and responds quickly to management change. So it's a good one to kind of monitor in that particular space. And I guess the negative side of this is, depending on the soil type, an unknown amount of the total carbon can be recalcitrant. So labile carbon may be a better indicator. The next one here, where we talk about microbial biomass carbon, generally related to soil microbiological activity and responds faster to management change than does soil organic carbon. I guess the issue here is, the different measurement techniques, and it is pretty hard to find labs that do this reliable, and requires, a pretty specialized equipment to really do that sort of stuff. Soil respiration rate measures the level of carbon dioxide generation indicating biological activity. The substrate-induced respiration test as a surrogate for micro biological biomass carbon. You've got to be aware though that soil respiration rate is highly variable and fluctuates according to the substrate type, moisture, temperature, and it must be measured under standard conditions. Other ones that I see commonly used in industry looking at this fungi to bacteria ratio. And, I guess it indicates the balance between the slower growing fungi and the faster growing bacteria. And it's influenced by the type of organic residues that are in the soil. the negatives to this are really that it's a highly variable with time and environmental conditions, and not specifically related to soil function affecting vine growth. The next one, potentially mineralizable nitrogen, measures the capacity of the soil to supply nitrogen over time, and it is related to the soil carbon-nitrogen ratio. No real negatives, but it's really hard to find, I guess, labs out there that can actually measure this over time. There are some different variants there, so just be aware of that particular issue as well. The next one is looking at enzyme activity. Looking at different enzymes that might be related to microbial activity. It's certainly easy to measure and responds rapidly to management changes. However, some of these measures potential not actual activity in soil, and generic enzyme tests not indicative of a specific soil function necessarily either. So again, just be aware of some of those problems with that particular way of assessing biological health. The last two here, really, we're stepping into the world of new genomic technologies that are able to, really take a sample of soil and assess the microbial diversity through, DNA, RNA testing, and offer, pretty good resolution and insights into the microbial community composition. In terms of some of the negatives around that at the moment, I guess we're just still learning a lot about these different measurement techniques and what they're telling us. So they can certainly give us an idea of that microbial diversity very quickly, but we're probably still lacking a lot of information that relates back to the function of that, or the presence of those different microbial communities and the function that they then represent in soil in relation to soil biological health. Last one, which we see obviously is being used quite a bit, is just the soil fauna. So the measurements of earthworms, nematodes, and protozoa. Certainly indicative of carbon cycling in the soil and nutrient turnover, and linking back to the soil food web. The negatives though is it's temporally and spatially variable, and nematodes really require someone who can identify accurately down to the species level. And again, some of these issues are not related to specific soil functions and can be very time consuming in terms of that approach. And that's why I guess some of these DNA approaches are really becoming a lot more favourable in that space at the moment too. So again, I might pause there. I've only got Bob maybe for about a minute or two here before he has to disappear. So just to ask Bob if he had anything else to add at that particular point there.
Yeah, just a couple of general comments, Mark. Again, one can tell a lot about the soil biological activity by visual observation of the soil in the field. And because most of the activity, biological activity anyway, is taking place in the surface soil, in the topsoil, the top 15 to 20 centimetres, and you can get a qualitative idea of the activity of earthworms, for example, and other forms of insects that decompose organic matter. So that's one thing you can do. But of the laboratory tests, the one that I would emphasize is soil organic carbon. And I'd add to that labile carbon, which is the carbon which is easily decomposable and is measured by extraction in dilute potassium permanganate or hot water. And the critical ratio there for a healthy biological function is to have a ratio of, at least 1% of labile carbon relative to the total soil organic carbon. And that's quite a useful indicator, which is pretty sensitive to change. And just touching on the fungi to bacteria, I think, again, this is something which is relatively easy to measure and does indicate whether you're getting the right sort of residues returned to the soil, which encourage fungal growth. And fungi are pretty useful organisms for improving soil structure at the micro level. So those are the 2 points I would make, Mark.
I'm just mindful of the time, Bob. Are you happy to take one question before you have
Sure.
to head off? Yeah, so we've got a question around stimulating mycorrhizal fungi in sandy soils. And also the follow-up is, are there any measures that can be taken to improve or grow a better soil health?
Well, taking the first one, mycorrhizal spores for the arbuscular mycorrhizas, which are the common ones infecting grapevines, they're very widespread. So if you provide a good source of organic matter into the soil through mulches, compost and things like that, then you should encourage these mycorrhiza to develop. One thing about mycorrhiza, however, is that they do tend to be suppressed if you've got too much available phosphorus, for example, or too much mineral nitrogen in the soil. So they tend to thrive better in soils that are not oversupplied with phosphorus or mineral nitrogen. But I mean, as a general rule, my take-home message is that build up the soil organic matter to organic carbon levels greater than 2%, and you can do this particularly in the vine row with compost, which is can be straw composted with chicken manure, for example, or pine bark composted with chicken manure. And this is one good way of increasing soil organic matter, particularly in the vine row. Of course, for improving soil organic matter in the mid rows, then cover crops are invaluable.
Okay, fantastic. Thanks for your further insights there, Bob. I'm aware that you've got some other commitments, so we'll let you go from here. And thank you again for joining.
Okay, Michael and Mark, that's good. I'll have to take my leave.
Again, appreciate your time. Mark, we'll hand back over to you.
Thank you. So moving on, so that sort of concludes the part talking a bit about which of the, I guess, the different attributes that we're looking to measure. But I guess I just want to really put some warning notes out there in terms of when it comes to interpreting this analytical data. The first one would be really understanding the units of measure. So you can send your soil off to a range of different labs and you'll get different reports back. I guess it's really making sure that we understand which units that they're being measured in. Now, ideally, a lot of those units should be in SI units, so standard international scientifically agreed units. So that way, when you're comparing, results between different labs or different sites or whatever it may be, that you're actually comparing apples with apples. So that's one of the common mistakes that we see around making sure that people understand that. The other one is that often many of these analytical tests that you're submitting your soil samples to, a lot of these tests have a limit of detection. And what do we mean by that? And it's really defined as the lowest quantity or concentration that can be reliably detected analytically. So most analytical labs really should provide you with information back to their clients in relation to knowing whether things are either not detected or they're trace and therefore indistinguishable from background levels. So it's important to, again, understand, you might be sending a sample in for being tested, but they're not able to really give you a number back because it's not giving you any number above a baseline level. The last point, which again may sound obvious, but it's really understanding the accuracy and precision of the analytical technique that you're employing there too. So, and I've got the set of bullseyes here on the right-hand side of the slide, really just to help understand. So, we all think that we're in a situation where we're in the top right-hand part of that quadrant where, I'm sending my sample in and everything that I'm sending in, I've got high accuracy and high precision. So it's just important to understand that because, if you were to send the same soil sample in to the same lab 10 times, you get an idea of where this is sitting in terms of that sort of stuff. So really understanding, and a lot of these, a range of different methods have quite varying levels of accuracy and precision. So it's important to understand that, and it has implications for the repeatability of the measures you're looking to take. Okay, next issue is, just around interpretation approaches. So again, we're getting numbers back from the lab. What do they mean? How do we interpret them? So what we're talking about here is just, a couple of three pretty common concepts around how we approach this. So the first one is interpretation around thinking or understanding what the optimum range is, and that is in relation to the most favourable level of growth, productivity or quality that we're likely to see at a certain concentration. So I've just given you an example here of potentially mineralisable nitrogen, and you can see I've just got the arrows there indicating, well, fruit yield is optimal between those particular ranges. So, again, it's important in understanding, again, the interpretation of the data that we're looking for here. The second concept is threshold, and really it's around the maximum concentration of a chemical attribute allowable for repeated exposure without producing an adverse vine health effect. And what I've tried to do to illustrate this point is look at the root zone salinity in grapes of own-rooted vines here. And what you see is beyond around about 2 decisiemens per metre in average grape root zone salinity, you see that the yield starts to decline from that particular point there. And this is, again, own-rooted grapevines, not rootstock. Rootstock is a bit more resistant to it. So again, that gives us a threshold. So we understand that, in soils where we're measuring soil salinity above 2 decisiemens per metre, that we're going to, be seeing potential problems with own-rooted Vitis vinifera. The last one, and which is used quite a bit too, is a benchmark. And a benchmark is a standard or point of reference against which things can be compared. And I've got an example here taken from work that was done by Jackie Edwards and Ian Porter out of McLaren Vale, and really looking just at soil pH within under vine here. And what you can see is there's quite a range. So this is looking at many sites within McLaren Vale. And at different points, you've got undervine, mid-row, and in the native areas around vineyards. And you can see quite a range in measures across McLaren Vale. But, you can take your result and sort of compare where you fit on that sort of, population, if you want, of results. So we find that's also useful in understanding where you fit, if you're within the normal range or you're at the extremes. The other one it's important to look at is, moving away from some of those sorts of interpretation issues, is looking at opportunities around, what are some of the minimum data sets that people might collect that are important in understanding soil health as well. And again, this is work that was derived from a large collaborative project that was led through the Victorian Government Department of Prime Industry through Jackie Edwards, Ian Porter, but also involved researchers here in South Australia in terms of Rob Bramley and co. But really try to say, well, what are some of these, biological, physical, and chemical properties that we should be really measuring, and that it should be, and then monitoring those over time to see how things change as an indicator of soil health. And these are some of the ones that they come up with, and as a way of kind of really assessing the overall health of the soil. So in terms of some of the biological stuff that you can see, microbial biomass, potentially mineralisable nitrogen and labile carbon, things we've talked about prior. Some of the physical stuff, aggregate stability and dispersion test, the AWRI test, aggregate stability and slaking test, and the air-dry soil consistence test. So some of the physical ones, and chemical, you can see here pH, electrical conductivity, cation exchange capacity, and total organic carbon and chloride. So those, this was, I guess, the digestion of a lot of thinking by a lot of different groups into what are some of the things that they should be really looking to monitor over time to, again, assess the health of a soil over time. Sort of moving into, when at the different stages, when we're looking to establish a new vineyard, obviously the choice of a new site depends on many things. It really depends on the climate, the soils, the genetics you're looking to introduce onto the site, variety, rootstock and clone. what style of wine you're looking for, what's your economic outcome. So those are the bigger picture issues about this. And also, obviously, issues to do with topography, the vigor potential of the site, the sun exposure and the solar path. What sort of row orientation you're looking to put in here, trellising and canopy management. Your water availability, both rainfall and likely supplementary irrigation, and the type of machinery that you need to move, and do you need to remove some of those existing soil constraints prior to planting. I guess the key thing there is obviously when you're at that new site thing, you really are going back to basics and really looking at trying to characterize some of those soil texture issues, some of the things that the inherent properties of the site, and looking at opportunities at major interventions, in terms of deep ripping, installation of drains, incorporation of gypsum or lime, reactive phosphate rock, or organic matter. And it's just much easier to really focus on that prior to putting a vineyard, putting posts and putting vines in the ground and putting all that infrastructure in place. Interestingly, there's not a lot published about, what are the soil macro and micronutrient levels that should be there, in a pre-plant situation. There's a bit of work that's been published by Dean Lanyon here that gives you a table there of, what are the concentration ranges of the key nutrients that we should be seeing there from a, marginal, adequate, high, etc. Level. I won't go through all those, but, that's there as a general indicator of what the sort of soil nutrient level should be for those particular nutrients at a, when establishing a new vineyard. In relation to when you come to looking at monitoring an established vineyard, really a lot of this, pushes back. Yes, you can certainly use soil analysis on a regular basis in this particular situation, but you should also really be looking at plant tissue analysis. And this is really important, probably more for the micronutrients, because we find that relevant soil tests are based on empirical extraction methods and not necessarily well correlated with vine nutrient status. So obviously what the vine is seeing is a synthesis of everything, where the roots are going and what nutrients have been taking up. So understanding plant nutrient status and whether you're assessing pedioles, lamina at different stages, etc., but comparing those back to standards. Also looking carefully for your visual symptoms of nutrient deficiency or toxicity or other problems as well in the vineyard caused by pest and disease. I think really important to understand all those things there. So I've just put up some different images of nitrogen deficiency, magnesium deficiency, iron deficiency, potassium and phosphorus deficiency, and also salt toxicity. And a lot of the plant tissue analysis, is done on pedioles collected at flowering, and according to the protocols that are described by Nicholas and White, and then those are compared to the Australian standards that have been sort of synthesised again from data from California and other work here in Australia. And just to remind you, yes, we have those, and there they are, the nutrient standards for pedioles at flowering. I'm not going to go through all those, but just to say that again, when we're collecting our pedioles, sending them off for analysis, we are comparing it to this sort of data to again understand where those optimal levels are for those particular nutrients and make sure that we're not seeing any toxicity or deficiency symptoms in there, and making the necessary management adjustments in the vineyard. Okay, looking at the final one here in terms of choices when reworking or replanting an existing vineyard. Basically this follows the same principles, but there's also the understanding that there's obviously a bit of prehistory here with prior history with other vines being in that place, and understanding whether they're susceptible to different diseases, whether there's poor winemaking for performance or other soil problems that really need to be considered. There's obviously an opportunity to ameliorate those soil problems, whether it's putting in drains or putting on more gypsum or putting in more organic matter. Or whether it's been such a poor performer, whether you actually then just do not replant that particular area of that particular vineyard. Also, if there's an aim to replant with new varieties, that opportunity to obviously really think about the choice of rootstocks here. And if you're in an area, and I know for example the Yarra Valley is going through a major replant, they're looking at phylloxera-resistant rootstocks. Or if I'm in the Sunraysia and I'm in a replant situation, I'm looking for rootstocks that are going to give me more resistance to parasitic nematodes. And also, obviously rootstocks do confer a level of inherent vigor, and give you obviously tolerance to other things like soil pH and also drought or salinity. So real good opportunity to rethink, your genetics and the tolerance to different soil-borne problems as well in that particular replant situation. And also, it should also be considered in any new planting situation as well. So look, just to summarise and wrap up, soil health can be assessed using, either, like I said, direct or indirect methods. Direct methods of assessing soil health rely on the measurement of soil properties and/or a plant tissue analysis approach. Approaches to assessing soil physical, chemical, and biological properties and the pros and cons have been discussed in a fair bit of detail here. Representative sampling, monitoring your soils over time, selection of reliable laboratories and good methods, and understanding units of measure and accuracy and precision issues. I think we've discussed those in a fair bit of detail. I tried to introduce to you today, and again, it's a summary of a fair bit of work that's been done a little bit of time ago now, probably about 5 years ago, but the concept of this minimum data set of those physical, chemical, and biological attributes that you might consider measuring on a routine basis. I guess, and the other thing is there's no one size fits all. The choice of soil attributes to measure will depend on a vineyard's circumstances as to whether you're looking to establish a new vineyard or whether you're looking at monitoring the existing or re-establishing a new vineyard as well. So probably summarises the technical parts of the presentation. I do want to acknowledge, obviously, it's pretty good having backup with Bob White on the line, help answer some of those questions that were being highlighted before. I really just wanted to thank, the likes of Jennifer Maguire, who helped me with a couple of the images here around nutrient deficiency, Ian Porter, who's obviously done a lot of work over the years with me in that area too, looking at setting benchmarks and recommendations for the management of soil health in Australian viticulture. And that final report's available on the Wine Australia website. Marty Longbottom for just the information around Sustainable Wine Growing Australia workbooks that I put there. And Con and Michael for their technical support here, putting the webinar materials together. And also Anne Lord, who's over there too, I can see. So thank you very much to all of the AWRI team. And just, for reference, I know people can get those, but just any of the references that I talked about through the presentation are there and available as well. So I'll stop there. Happy to take questions.
Fantastic. Thanks for your presentation there, Mark. Mark will stick around for a little while. So if you've got any additional questions, please start sending them through. Just a reminder, open the Q&A button on your webinar toolbar and click to send it through. So we did have a question come through a bit earlier, Mark, and it was around carbon. I know you touched on this briefly. What are your thoughts on using carbon as an overall measure of soil health? And does soil organic carbon do this, or do we need to add another measure?
Yeah, it's a good question. I think, and I sort of wish Bob was here to help answer the more technical elements here around that, but I think measuring soil carbon is really important. Organic matter, soil carbon, all those things are really important. I know that the Cornell soil health assessment platforms really rely on the measurement of soil organic carbon as well. So yeah, I think it's a really important measure to make sure we've got incorporated. And it's probably, as I've thrown all of these possible measures at you today, the real next step you'll find is when you go to find a lab that can actually measure these different things. You don't want to go to 7 different labs. You want to be able to go to one that can kind of put them together a bit for you. And that's part of the challenge that I find with, which lab do you pick and which one do you go to? They can offer the range that you're looking for. But yeah, just to answer the question that, if you've got a lab that can measure that, that's a great way of getting an assessment of soil carbon and soil and a bit of a surrogate for that sort of soil biological health as well.
Thanks for your question there, Mark. Another one here about, apart from soil deficiencies, is there any work that correlates soil health with wine quality?
I see that's come from a friend of mine in Coonawarra. Thank you. And I just wanted to say, look, it's a really good question. And we, in the book that we published, we tried to go into that in the later chapters. There actually isn't a lot of good links, apart from, say, high salinity and those sorts of things, which we know causes adverse problems with vine health and so forth. There's not a lot of things that linking, say, nutrient levels in soils to problems in the vine, and sorry, links through to wine quality. Again, except for things like salt problems and the toxicity problems that causes. So yeah, it's a good question, and what we try to articulate later in the book is really understanding that the soil infers inherent vigor on the vine, and that's really important for, I guess, vine balance and the overall architecture of the vine and bunch microclimate, which is really important then for the expression of quality in those particular situations. So I've tried not to use the T word today specifically, and I won't utter that, but I think it's a good question, and we go into that in a little bit more detail later in the book.
Okay, thanks for your question there, Pete. Another one from Andrew. Bob mentioned the benefits of overcropping, Mark. At this time of year, what are your thoughts on either rolling or slash side throw undervine? Auobviously,
again, I saw the name of the person here, I won't name, but I see that they're obviously from a higher rainfall zone in Australia at the moment. Yeah, it's a good question. I think the rolling concept, there seems to be a bit more of a favourite. That seems to be being more favoured at the moment, because I think it just preserves that biological material a little bit more. I think once you're trying to put that back into the soil, you're going to turn that organic matter over pretty quickly. And so therefore, and basically you're providing food for the microbes a lot quicker to really metabolize, and I think the lifespan of that organic matter is a lot less. Where you're rolling it, I think you got the benefits of it being a bit slower to decompose, and then you've got a lot of the other benefits in terms of probably restricting a bit of that reflection of the radiant heat back into the canopy, and providing problems with a bit more heat stress in vines as well.
Okay, thanks for your question there, Andrew. That looks like it may well be it with regard to the Q&A, Mark. Do you have any final comments before we start to wrap the session up?
From me, no, I'm right. Thank you. I just thank you for the opportunity to present today, and I hope people enjoyed it. And yeah, if there are any other questions, feel free to contact me outside of the webinar.
Yeah, fantastic. Thanks very much, Mark, also for coming in and presenting and providing the audience some really key learnings around this important topic. Mark, would you mind just slipping through to the final slide there? There's one at the end. I'd also like to extend a thank you to Bob. He's not in the session now, but thank you again, Bob, for joining the session. Appreciate your input. And also a thank you to the audience for listening in and participating. All attendees will receive a link to view the recording of this session tomorrow. The next AWRI webinar, which as I mentioned at the start is the last for the year, is on Thursday, the 5th of December. Darren Ray from the Bureau of Meteorology will provide a seasonal outlook for vintage 2020. So if you haven't registered for this session and you would like to, please visit the AWRI website. Thank you again, and I look forward to seeing you again at the next AWRI webinar.